Cell culture is a laboratory technique where living cells are grown outside of their natural environment, in controlled conditions, usually inside flasks, dishes, or bioreactors. Instead of cells being inside a body, they are maintained in a nutrient rich liquid called culture medium, which provides the food, vitamins, minerals, and growth factors the cells need to survive and multiply. This allows scientists to study how cells function, how diseases work, and how drugs affect cells, all without needing to experiment directly on living organisms at every step. In simple terms, cell culture is the process of keeping cells alive and growing them in the lab, almost like farming cells. Cell culture is one of the most essential techniques in modern biology and medicine. Here's why: studying diseases. Scientists can watch how cells behave when they are infected, stressed, or genetically altered. This helps us understand how diseases form and progress. Testing drugs safely. Before a drug is given to animals or humans, it is first tested on cultured cells to see if it works and to check for toxicity. Producing medicines and vaccines. Some vaccines and therapeutic proteins, like insulin, are grown using cultured cells. Tissue engineering and regenerative medicine. Cells can be grown into tissues or even organ-like structures to help repair damaged body parts. Basic research. Cell culture allows scientists to understand how cells divide, signal, differentiate, and respond to their environment. Without cell culture, many advancements in medicine and biotechnology simply would not be possible. Just like plants need soil, water, and sunlight, cells need specific conditions, nutrients, provided by the culture medium, warmth, usually around 37 degrees Celsius, a sterile environment, proper gas levels, and stable pH. To maintain these conditions, cells are kept inside specialized devices called incubators. Cells used in culture can come from various sources: human or animal tissues, stem cells, and immortalized cell lines. Different cell types require different nutrients and conditions because each has its own biological needs. Types of cell cultures. Primary cell culture, taken directly from tissue and grown for the first time. They closely resemble real cells in the body, but they don't survive or divide forever. Cell lines. Cells that have adapted to grow continuously. They are easy to culture and widely used in research, but they may differ slightly from normal cells. 3D cultures and organoids. Instead of spreading cells flat on a dish, cells are grown in 3D shapes that mimic many organs. These structures behave more like real tissues in the body. How cell culture is done. Cells are placed in a sterile dish or flask. Culture medium is added to provide nutrients. The flask is placed in a warm incubator with controlled humidity and gas levels. As cells grow, scientists monitor them under a microscope. If they grow too densely, the cells are split into new dishes to prevent overcrowding. Medium is changed regularly to remove waste products. It's a careful, consistent, and clean process, much like caring for a garden. The biggest threat: contamination. Because cells grow in nutrient-rich solutions, bacteria, yeast, and fungi love to grow there too. If contamination occurs, the entire culture can be ruined. To prevent this, scientists use sterile gloves, clean airflow hoods, disinfected tools, antibiotics in some cases, and careful handling procedures. Good technique is critical. Cell culture requires patience and cleanliness. Applications in real life: cancer research, vaccine development, cosmetics testing, drug discovery, and gene editing research. Cell culture acts as the bridge between molecular research and real-world medical use. In summary, cell culture is the technique of growing cells outside the body in controlled lab conditions. It provides a controlled environment for studying how cells behave, developing new medicines, testing treatments, and producing biological products. It forms the foundation of much of modern biomedical science and biotechnology. Put simply, cell culture lets scientists grow and study life at its smallest scale, giving us the tools to understand disease, develop new therapies, and push the boundaries of medicine.